You can see your screen?
Yes, we can see your screen.
Yes. Okay. So thank you, Smitesh. So today we are going to present the use case 3A that is about EMSO-Azores Marine Species Detection Service, and we'll share the presentation between myself, Catherine Bourmans, and Vanessa Tosello. So I'll start with just to remind what is the use case 3 about. So it's based on the video imagery collected at EMSO sites for ecosystem monitoring. So for people who don't know EMSO, it's research infrastructure in Europe, networking different observatories at different locations. And each observatory is composed of different instruments measuring geophysical, biological, physical, and different other parameters. And on several sites, cameras are also integrated in the observatories. So the objective of the use case 3 is to work together between the 3 sites of EMSO to develop an integrated service for automatic analysis of video collected at those sites. And the main objectives are fauna and habitats identification and monitoring. So the 3 sites are EMSO-Opsea, EMSO-Azores, and EMSO Smart Bay. So for our use case on EMSO-Azores, we use data from the ecological module Tempo. That's a module installed on the 2 observatories, one on the mid-Atlantic ridge and the other one in the Pacific. Each modules collect video since 2010 and 2011 for the one in the Pacific. They record videos at a precise frequency. So during 2 minutes each 6 hours throughout the year for the EMSO-Azores one, and during 20 minutes each 4 hours for the Pacific one. And just to precise, those modules are deployed in ecosystems of hydrothermal vents. So that's an example of the image we collect in EMSO-Azores. So it's a fixed camera in front of a mussel assemblage, and we monitor all the fauna that is living there. And that's an example from the Pacific ecosystem. So the biodiversity is different. And for this observatory, we also have a zoom for the video at a certain frequency also. So the video that are collected are then annotated through a citizen science project that is called Deep Sea Spy. It's a web platform allowing citizens to annotate images that we extract from the video. We propose a list of species to detect and identify, and it allows to build a data set of annotation for those images. So we use the first mission annotation mission data set for the purpose of the use case 3E. So it's about 4,000 images, 15 species to identify, and it produced 250,000 annotations. So to give you more details about this data set, so you can see it's a quite unbalanced data set in terms of classes. So we decided to focus on the most annotated species, that is the buccinid, the snail. It's the class for which we have the most annotation, and it also has the advantage to be quite easy to recognize because it's a clear object on the dark background. But we also use another class to develop the use case. It's the Bitograide class, so they are crabs. We have really less annotation for this class, but it's visible on another type of images, and it has also the advantage to be quite a clear organism to identify on images. So it's, even if we have less annotation, we expect it to be more easily recognizable.
Okay. So now I'm going to present the pipeline that we implemented in the frame of this use case. As Catherine said previously, we use citizen data from DeepSea Spy platform to train AI model in order to recognize automatically deep sea species in image collected from seafloor observatory. So we have implemented a pipeline which includes several steps from data pre-processing to standardize and clean and validate the annotation, and to prepare the data for training an AI model. And finally, we train a model, and the final pipeline was deployed on the iMagine platform. Preparing the training dataset was quite challenging due to the diversity of species and the variable methods used for labeling the data. Each image was annotated by 10 people, leading to variation in accuracy and consistency. And also, the different species were labeled using different formats, like point, line, and polygons. So we had a lot of cleaning and standardization to make the data usable by an AI model. So here is an example of the first step, which consists of conversion of various annotation types to bounding box. Some correction have been applied to compensate potential conversion error, and adjustments have been made depending on the initial annotation and the species. For the point, for example, we had a padding that depended on the species. And for the line, we took into account the slope of the line to avoid having bounding box that were too flat and which didn't cover the specimen properly. The next step was unification of overlapping bounding box. As images have been annotated by several citizens, we decided to unify the annotation, and to do that we use a program based on intersection over union, which is a metric that measures how much two bounding boxes overlap. And we only boxes with a mouse overlap were merged, and single boxes are were discarded because considered as misinterpretation. And the merging of bounding boxes depends on both the number of available annotation and their overlap percentage. Here you can see here that now we only have one bounding box per specimen. Here again on a zoom image. The next step include a visualization function that lets you display your images with bounding boxes overlaid. This is very useful for checking whether the annotation are correct and if the cleaning process worked as expected. Maybe it's too small to see, but here we can see the bounding box in red on each images. The next step is a verification step to ensure our data is the cleanest possible. And we provide a function to generate thumbnail from images and bounding boxes. This function allows user to visually inspect and verify the data quality, and they can choose to remove some thumbnails which are not relevant, and the function will take into account these changes, and the non-relevant thumbnail will not use in the model training. Next, the pipeline will provide a set of tools to prepare automatically the dataset for YOLO V8 training. It consists of conversion of the bounding box into the YOLO format, which is shown here. We have a tool also which prepares the split in a train val test folder, and we can generate also automatically YAML configuration file, which is expected by YOLO to locate the data and know the different classes available in the dataset. So next, we trained the YOLO V8 model, which is a convolutional neural network-based object detection model with fast processing of images, fast learning, and high accuracy. He has the ability to detect small objects in underwater video imagery. He has a robust performance for analyzing various marine species. That's why we decided to use this model. We train this model on two particular species presented previously by Catherine, so buccinid snail and beetle-grade crab. We used citizen annotation to train the model, but we used expert annotation to validate the model when expert annotation was available, in order to answer better performance. All the data is available on Sinoe. Sinoe is a French data repository, and Zenodo. So we have published the raw data and images, the training data set, and the pre-trained model. And the code is also available. The full pipeline is available on GitHub and on Docker Hub. So the pipeline can be run using a Jupyter Notebook for an interactive mode or in a batch mode execution. You can do training and prediction using the DPass API, and you can use the pipeline locally or in the iMagine platform, and everything is well documented in a README file on GitHub and on the platform. Just very briefly, if you want to use the pipeline locally, you can do it from the GitHub repository. I don't want to detail this this slide, but if some of you are interested, you can follow the different steps and use it locally. Not that this solution need to install a Python environment. The second option is to use the pipeline locally using the Docker image. In this solution, there is no need to install Python environment because the Python environment is included in the Docker image. I don't want to detail this line, but you can follow all the steps to run the pipeline using the DPass API or the Jupyter Lab. And you can, of course, use the pipeline on the iMagine platform, which provide computing resources to run training and inference. You will not need to install Python environment, and you will be able to use the pipeline with DPass API in command line in Jupyter Lab. You will be able also to link your deployment on the platform to a Nextcloud repository, and you will be able also to download external dataset on your Nextcloud repository to use it directly in the platform. So I'm going now to show you how to use the pipeline in the iMagine platform. Due to the power outage in Spain and Portugal, the iMagine platform has been inaccessible for the past 2 days, which made difficult to fully prepare this webinar. So as a result, we have decided not to make a live demo today and just show you a snapshot. So I apologize for the changes, but I believe that this is still valuable for user, as the presentation will be available on the iMagine website after the webinar and can serve as a user guide. So we can go now on the iMagine marketplace, and you can select deep species detection model. Here you will find some documentation about the module. and you will find all the link to access the dataset in Zenodo, in Sinway, the code in GitHub, and the Docker image in Docker Hub. You can access these resources without login. If you want to use the module in the platform, you will need to log in. And if you don't have an account to log in, you can send access request on the iMagine website. And you will have to fill in a form to request access. So then when you are logged in the platform, you can access an how-to button. The first one, it's a link to use the platform using a short 10 minutes deployment. I will not explain this part. The other option is to use a deploy button here, and then you have to select Inference API plus UI option. Then you will have to enter some information like the title and name of your deployment. You will have to choose which kind of service you want to deploy. Is it a DPaaS API or Jupyter Lab? For Jupyter, you will have to enter a password, and this password will be requested when you will access the deployment. So I enter a name for my deployment and click on next button. Here you can define the computing resources you need, the number of CPU, number of GPU, and the RAM and disk memory required. And in the next tab, you will have the possibility to link your deployment to your Nextcloud account. I have included a link to explain how to fill in the different fields of these forms. Note that this link can be done only once for all your deployments. And the next step is here. You can ask for using external dataset by clicking on the Add Dataset button. And here we can search in a dataset in reference in Zenodo. So here I choose our dataset, and I click on Add button to add it. And I select another one, also another dataset here. And next, that's all for ask for a deployment. And you can click on Submit button at the bottom of the web page. And just before continuing, when you ask for download the external dataset, the datasets will be saved in your Nextcloud workspace in A4S storage and dataset folder. So now we can go in the deployment menu to access the deployment that we have just created. And you will have to click on the quick access button to to access the deployment. I'm going to show you two kinds of deployment, one using DPass API, and after that I will show you deployment using Jupyter. So when you run the DPass deployment, a window like that will open, and you will be able to access different possible action, like here you can retrain a YOLO V8 model with your own data. I will not detail this part, but you can also make some prediction using our pre-trained models on Bixinid snail and Vitograde crab. So if you click on this menu, you will access this information. You will have to click on Try It Out button on the top. And here you will be able to browse and select an image. I choose an image with Bixinid snails. Here you will be able to choose between our two pre-trained models, one for the snails and one for the crab. Here I keep the snails one. And you will have to click on Execute button at the bottom. And the results of the prediction will be available here in JSON format. So you can see that we have a lot of Bixinid snail on this image. Now I'm going to show you a deployment using Jupyter. When you click on Quick Access to the deployment, you will arrive in a Jupyter lab, and we will be able to open the Deep Sea Lab notebook, which is available under Deep Species Detection and Deep Sea Lab folder. The first part of this notebook asks to define the path to your data. The first pass is a pass to your annotation in a CSV format. The second one is the folder containing the input images. And the last one is the path to the directory where the output result will be saved. If you have a Nextcloud account and if you included the link in your deployment to your Nextcloud workspace, you will be able to define a path using a storage directory, which correspond to your Nextcloud workspace. So the next step is you have the possibility to display the data set. Next step is the conversion of the different kind of annotation, point, line, and polygon to bounding box. At the end, you have the possibility to save the new data set with the bounding box. And the saved file will appear in the save folder you defined previously. The next step is visualization. So you can ask the pipeline to visualize your images with bounding boxes. In this case, the function will create a stock repository with all the images, and you can navigate into. You can see here that the bounding box are not unified. So the next step is to unify the bounding box. Here you will find several cells, and you will be able to choose between that cells depending on how do you want to unify the bounding box. And at the end of the unification, you can also save a new CSV file with the unified bounding boxes. You can now run a new visualization to see the new bounding boxes, and you can see here that we have only one bounding box per specimen. The next step is a catalog of thumbnails, so we can ask the pipeline to create a new folder called catalog to save all the thumbnail, and you will be able to select the thumbnail you want to keep or not. The next step will prepare the data for YOLO. So the first cell converts bounding box in a YOLO bounding box. And the second cell will split the dataset in a train val test folder. Here we are creating a YAML file to expected by YOLO V8, and this is the first cell here. The next cell is just loading a YOLO model, and here you can train the model, and the result of the training will be saved in the runs folder here. And finally, you can make a prediction directly in the notebook. You have first to load the train model. Here I took our pre-trained model on Vexini class. And here you can define a folder with the images to predict. And here you will ask to prediction. The prediction will be saved in a new folder, inference results. And so we will find this kind of results here. So my demo is almost finished. So now some perspective and application concerning the model development. We plan to do some code optimization, especially in the pre-processing pipeline, to reduce time responses. We would like also to improve the model performance on buccinid snail and beetle-grade crab by introducing new data in the training dataset. And we would like also to train the model on other species, of course. So I give the floor to Catherine for the scientific applications.
So yes, at the end, thanks to the development carried out during this use case, we are able to produce automatically identification of species on a long-term video collected on the observatory. So obviously we can produce temporal analysis on species population, and in relation with the parameters we can record on observatories, also on a long-term series. And more generally, we can study distribution and evolution of species, abundance and biology, not only in time, but also in space, because the development can be applied also to video or images collected spatially. And lastly, the data processing pipeline has to be the advantage to be able to be applied to any kind of imagery that can be collected by fixed cameras or especially by mobile devices for C4 mapping. That's it for our presentation, and we